Hello guys, welcome back to my channel again. So today in this lecture we are going to talk about polymerase chain reaction, which is also called as PCR. So basically what is PCR? So PCR is an artificial method or artificial technique to replicate the DNA in a laboratory condition. So what are the various components you need to do a PCR? So PCR is to amplify the DNA to make number of copies, multiple copies of the same sequence that you know from a fragment of DNA. So you need DNA sample, you need primers, one forward primer and one reverse primer, and you need nucleotide base pairs. And apart from all these three components, you also need an enzyme which is called as a replicating enzyme. Generally in PCR, take polymerase is used. Take polymerase is a kind of enzyme which do not get denatured at 95 degrees Celsius because all the other protein gets denatured and its function get losses at some temperature when the temperature increases. So take polymerase was taken from Thermus equatis, which is an archaea, hot spring archaea or bacteria. Apart from this, you also need take or buffers, take buffer, and you need PCR tubes and one thermocycler machine or PCR machine. So all these are the basic component of a PCR, and you need to do or to make a reaction of PCR. So this mix buffer is also called as master mix. So these are the various components of a PCR what we need. These all things are needed to perform a PCR reaction. So the PCR cycle in one cycle has multiple steps. So at 95 degrees Celsius, the DNA strands get denatured. It gets separate from one another because DNA is double stranded. So the DNA gets separate, our sample DNA. And at almost 55 degrees Celsius, that is annealing, the primers get bind to the templates, the forward primers and reverse primer. And at 72 degrees Celsius in the third step, the primer get extended and make the with the help of the enzyme, and it can replicate the DNA in both the strands from 5 days to 3 days. And so the extension and annealing temperature get changes or get varies from DNA to DNA. For example, from primer to primer, from gene to gene. If you are amplifying a gene, for example, if you are amplifying 16S rRNA gene, the primer for 16S rRNA, universal primer or species specific primer, the temperature for various annealing and extension temperature varies from one gene to another gene, from one organism to another organism. For example, if you are taking COI gene, if you are taking 16S rRNA gene, for both the genes, the temperature may varies from one organism to another organism, or it may varies from one gene to another gene. So thank you so much for watching this. So the lecture end here, and I hope that you get to learn about various components and the steps in a PCR cycle or in a thermocycler. Thank you so much.